Hey guys, Miss Cote here, and this is the last episode of the Energy Unit. We've looked at cell and animal metabolism and cellular respiration, and now we're going to spend some time going over the process of photosynthesis, how cells can capture light energy from the sun and convert it into chemical energy stored in organic molecules. But before we jump into the processes involved, we need to go over a few things. So let's get started. Photosynthesis occurs in plants, algae, certain unicellular eukaryotes, and some prokaryotes. It can occur both on the land and in the water. These organisms who have the ability to make their own food are called autotrophs or producers. Organisms that use light are more specifically called photoautotrophs, as opposed to chemoautotrophs. Those are organisms that use chemicals to make food. And the rest of us, the heterotrophs or consumers, are dependent on the food they make. Photosynthesis is basically what feeds the world. We just saw that there are other organisms that can perform photosynthesis, but in this chapter, our emphasis will be on plants. And while all of the green parts of a plant are capable of doing photosynthesis, leaves are where most of it happens. So we're going to focus on that. If we were to take a cross-section of a leaf, it would look like this. On the underneath of the leaves are microscopic pores called stomata. These pores are what allows the exchange of gases. Remember, carbon dioxide is a reactant, so it needs to enter the leaf, and oxygen is a product, so it exits the leaf. Also inside the leaf is the vascular system, or veins. But unlike our veins, which carry blood around our body, plant veins are either bringing water up from the roots to the leaves, or bringing the sugars that are made through photosynthesis to the roots and other parts of the plant that can't do photosynthesis. But what we want to focus on are the mesophyll cells, which is where photosynthesis takes place. Inside the mesophyll cells are where you find the chloroplasts, those organelles that we learned about in the cell unit. Let's review the parts of a chloroplast. A chloroplast is made of a double membrane, and inside is a fluid called the stroma. Also inside the chloroplast is a third membrane system, connected discs, called thylakoids. The thylakoids can be stacked, and one of these stacks is called a granum. The plural is grana. One very important component of the chloroplast is a pigment called chlorophyll. This is the molecule responsible for absorbing the light energy and also for giving the plant its green color. We'll discuss chlorophyll in more detail in a bit. Obviously, you need to know the equation for photosynthesis, and if you really look at it, hopefully you'll notice that if you switch the reactants and products, you would have the equation for cellular respiration. And not to spend too much time on this, since I covered this in the cellular respiration chapter, photosynthesis is also a redox process, where once again, it's basically the reverse of cellular respiration. Here in photosynthesis, water is being oxidized. See, it's lost its hydrogens and the electrons that go along with it, and the carbon dioxide is being reduced. It gains the gains the hydrogens and therefore the electrons. And while cellular respiration is an exergonic process, energy is released as a product of that one, photosynthesis is an endergonic process. So energy is a reactant. In this case, the energy comes from the sunlight. Before we jump into photosynthesis, let's do an overview. Photosynthesis occurs in two parts. The first part is known as the light reactions, the photo part of photosynthesis. The light reactions take place in the thylakoids inside the chloroplast. Here we get the process started with two of the reactants, light energy and water, and produce oxygen as a byproduct. In between, we're converting the energy from the sun into two different molecules. ATP is made through a process called photophosphorylation, and an electron carrier called NADP is reduced to NADPH. That's another relative of NADH. The second part of photosynthesis, the synthesis part, is called the Calvin cycle. The Calvin cycle takes place in the stroma, the fluid inside the chloroplast. Here is where the third reactant, carbon dioxide, comes into the process, and the sugar is produced at the end. The ATP and NADPH created from the light reactions are used to fix the carbon from carbon dioxide into sugar. Let's take a minute and look at sunlight and why it's so important in photosynthesis. Not to get too much into physics, but light is a form of electromagnetic energy, and it travels in waves. These wavelengths can be measured by taking the distance between the crests of the waves. Don't worry, this is not something we measure in this class. What you are looking at here is called the electromagnetic spectrum. And while there is a wide range of electromagnetic energy, in photosynthesis, we only concern ourselves with the visible light, which is a very small part. These are the colors that we see with our eyes. And as you can see, each color is associated with a certain wavelength, which you don't have to memorize, by the way. So what does this light do? The light, which actually consists of discrete particles called photons, is going to interact with molecules called pigments. Pigments have the ability to absorb photons of visible light. Pigments are specific to which wavelengths of light they can absorb, and whatever is not absorbed is reflected back. In the case of plants, I said earlier that an important molecule found in chloroplasts is the pigment chlorophyll. Chlorophyll absorbs mostly in the red and blue areas of the spectrum, but does not use green light. So the green light is reflected back at us, which is why plants appear green. These wavelengths can actually be measured using a machine called a spectrophotometer. There's a nice picture in your book that explains more and how it works. But the short version is, by measuring the absorbance of pigments, you can create an absorption spectrum. Here is an absorption spectrum for the main pigments involved in chlorophyll. If you look at the y-axis, the higher the peak, the more light is being absorbed. Chlorophyll A absorbs most light in the blue-violet and red wavelengths. Chlorophyll B absorbs in the blue and red-orange areas, and the carotenoids absorb best in the blue-violet areas. But also notice that there's basically no absorption in the green wavelengths, which is why plants appear green. If it's not being absorbed, it's being reflected, and we see what is reflected. The pigments in the thylakoids are grouped into complexes called photosystems. The photosystem is embedded in the thylakoid membrane, and here's how it works. The photons of light hit the pigment molecules in the photosystem. Each pigment molecule extracts the energy from whatever wavelength it's capturing the energy of the light from. Then all of these pigment molecules pass along the energy down the chain until all available energy reaches the main pair of chlorophyll A molecules. The energy excites a pair of chlorophyll A molecules, and they jump out of chlorophyll A and are caught by a primary electron acceptor. In the light reactions, there are two types of photosystems, photosystem 1 and photosystem 2. Now, not to confuse you, but while the photosystems were named in the order for when they were discovered, photosystem 2 actually comes first in the order of the light reactions. Let's look at the linear flow of electrons in the light reactions. Like we discussed in the last slide, a photon of light will hit a pigment molecule of photosystem 2, and that energy will be shuffled down the line until it reaches the main pair of chlorophyll A molecules, which cause a pair of electrons to become excited, jump out of chlorophyll A, and get picked up by the primary electron acceptor. Now, it's important to realize that the electrons that just jumped out of chlorophyll A need to be replaced, or the light reactions will eventually stop due to no more electrons to excite. Here's where water comes in. A water molecule is split into hydrogen ions, the H+, which will be used to create a proton gradient in the electron transport chain. Electrons, which are used to replace the electrons that were lost, and oxygen, which is released as a product of photosynthesis. All right, back to those excited electrons. After being picked up by the primary electron acceptor, the electrons enter the electron transport chain. Just like in cellular respiration, the electrons are passed down a series of proteins, and as they give off energy, that energy is harnessed to create the proton gradient, which is then used in chemiosmosis to make ATP through ATP synthase. Meanwhile, photons of light are also hitting photosystem 1. The pigments are absorbing the energy and passing it to the main chlorophyll molecules. This excites the electrons and causes them to jump out of photosystem 1 into another primary electron acceptor. The hole that's left from the loss of those electrons is filled by the electrons that originally came from photosystem 2. So there's no need to split water for this photosystem. And these electrons are now passed to another electron transport chain, but in this chain there's no proton gradient or chemiosmosis to make ATP. Instead, the electrons end up reducing NADP to NADPH through an enzyme called NADP reductase. So NADP is considered to be the final electron acceptor for photosynthesis, as opposed to oxygen being the final electron acceptor in the electron transport chain in cellular respiration. So as I said earlier, the purpose of the light reactions is to convert energy from the sunlight into ATP and NADPH, which we just did. And this energy will be needed for the Calvin cycle. Please understand that while both cellular respiration and photosynthesis contain this ATP making process called chemiosmosis, there are differences between them. First, the creation of ATP is done using different sources of energy. Cellular respiration uses the energy from organic molecules such as glucose, while in photosynthesis the energy comes from light. Obviously, this process occurs in different organelles in the cell, so there are differences between the spaces that are used. Take a look at the diagram and see in which directions and spaces the protons are pumped into and where they flow through ATP synthase. So to overview the light reactions, which takes place in the thylakoid membranes, this is how it's set up. As light hits the photosystems and the excited electrons move down the electron transport chain, ATP is made in the first part of the chain through ATP synthase, and NADPH is made at the end of the second chain. Both of these energy-containing molecules will then go out to the stroma for the Calvin cycle reactions. The second part of photosynthesis is the Calvin cycle. Here is where the glucose or other organic molecules are made. Carbon enters the cycle as carbon dioxide, another reactant in photosynthesis, and while it doesn't leave the cycle as glucose, it leaves as a 3-carbon compound called G3P, which can then be used to build the larger molecules. So to make one molecule of G3P, the cycle needs to turn 3 times, since it only fixes 1 carbon at a time. The Calvin cycle has 3 main phases to it. The first phase is where the carbon from carbon dioxide is fixed into an organic molecule. Here, an enzyme called rubisco is used to join the carbon from carbon dioxide to RUBP, a 5-carbon molecule. This produces an unstable 6-carbon molecule that very quickly breaks down into 3-carbon molecules. The second phase is the reduction phase. By reducing that 3-carbon molecule to another molecule called G3P, the energy is being transferred from ATP and NADPH into the G3P. One of the G3Ps will leave the cycle to become glucose or another organic molecule, and the rest of the G3P molecules will go through a series of steps to rearrange themselves to regenerate RUBP. This is referred to as the regeneration phase. The Calvin cycle would stop working and therefore making G3P if there's no RUBP. And then the cycle continues again. So if 1 carbon is fixed per turn, a molecule of glucose, which has 6 carbons, would need 6 turns of the cycle. And that wraps up photosynthesis. Plants can convert the energy from the sun to energy-containing organic molecules. These molecules can either be used by the plant to do cellular work or transferred to animals so they can do work. And I apologize if I'm talking a bit fast, but I was trying to get this under the 15-minute mark. So enjoy the rest of your day and be a good human.